This embryology highlight screencast deals with nervous system development and in particular CNS regions. Neuroepithelium of the neural tube gives rise to the central nervous system, which includes the spinal cord, the brain, and the optic nerve and retina, which grow out from the brain. Initially, the rostral end of the neural tube forms three vesicles, the forebrain, the midbrain, and the hindbrain, and caudal to these, neural tube becomes spinal cord. Subsequently, the forebrain divides into a telencephalon and a diencephalon. The telencephalon becomes the cerebrum, consisting of two cerebral hemispheres, and the diencephalon gives rise to the optic nerve and retina. The retina develops as an optic cup. The hindbrain gives rise to the metencephalon and the myelencephalon. The metencephalon becomes the pons and cerebellum. The myelencephalon is the medulla oblongata. These five regions, the telencephalon, the diencephalon, the mesencephalon, the metencephalon, and the myelencephalon, are used as convenient regions for categorizing brain anatomy. During development, the brain undergoes three flexures, which largely disappear in domestic quadrupeds because the head is aligned with the spinal column. In primates, the flexures are more important because the head is oriented at a right angle to the vertebral column. The spinal cord develops by bilateral growth of alar and basal plates with no growth of the roof plate and the floor plate. As a result, the spinal cord appears bilaterally symmetrical. The two halves of the spinal cord are separated by a ventral median fissure and ventrally and dorsally by a dorsal median sulcus and septum. The septum may become a fissure in lumbar segments. The neural cavity becomes a central canal which is lined by ependymal cells. It contains cerebrospinal fluid and communicates with the fourth ventricle of the hindbrain. The mantle layer gives rise to gray matter, which is butterfly-shaped or H-shaped in transverse sections. The gray matter is subdivided into a dorsal column, a ventral column, and between these an intermediate region. In transverse sections where the gray matter appears in profile, the columns are referred to as horns. So we have a dorsal horn and a ventral horn. And in thoracic segment, we also have a lateral horn, which is formed by preganglionic sympathetic neuron cell bodies. The marginal layer becomes the white matter, and it is divided bilaterally into funiculi. There is a dorsal funiculus, a lateral funiculus, and a ventral funiculus. The boundaries of the lateral funiculus are approximately where the dorsal root enters and the ventral root exits. Spinal cord segments that innervate limbs are enlarged. They are enlarged because they have more neurons, particularly more efferent neuron cell bodies in the ventral horn. The reason there are more neurons in enlargement segments is not because more neurons were generated in these segments. It is because fewer neurons degenerated in those enlargement segments. The limbs provide more target cells and more neurotrophic molecules, so more neurons survive during development in the segments that innervate limbs compared to segments where there are no limbs, such as this. The medulla oblongata and pons regions of the hindbrain, the brainstem of the hindbrain, give rise to the fourth ventricle. And this takes place when the alar plates move laterally and stretch the roof plate over the surface of an enlarged cavity which becomes the fourth ventricle. The term alar means wing, and you can see that the reason the dorsal plates are named alar is because they wing out laterally in the process of forming the fourth ventricle. The roof plate consists of ependymal cells covered by pia mater, and this combination gives rise to two choroid plexuses bilaterally in the roof of the fourth ventricle. The pia mater supplies vessels, and the ependymal cells supply the secretory cells of a choroid plexus, which produces cerebrospinal fluid. Here we see two levels through the brainstem. More caudally, the cavity looks like a central canal, but as these dorsal alar plates swing laterally, wing out, they stretch a roof plate over a large fourth ventricle, and the roof plate consists of ependymal cells which line the ventricle, covered by pia mater on the surface, and they proliferate into bilateral choroid plexuses. The choroid plexuses extend through a lateral foramen, and through these bilateral foramina, the cerebrospinal fluid gets out so it can surround the central nervous system. So the basal plate, which contains the efferent neurons, then has a more medial position as the alar plates swing laterally. So if we look at this cartoon in the spinal cord, we have the alar plate situated dorsal and the basal plate with the efferent neurons ventrally. And then as the alar plates swing out, alar plate neurons are lateral and the basal plate neurons are in a medial position. Another characteristic of the brainstem, is that the white and gray matter are more intermixed. They're not segregated like in the spinal cord. So, for example, here is some white matter, here's some gray matter, here's some gray matter, here's gray matter, here's white matter. And then there are areas where the white and gray are very much intermixed. We'll call this reticular formation in the future. There are several reasons for this mixing. One of the reasons is the cerebellum is developing above the hindbrain, and that creates a need for excess nuclei and tracts. This transection shows what a mature cerebellum looks like. It's sitting above the brainstem of the hindbrain. We see the fourth ventricle with bilateral choroid plexuses in it. The cerebellum features deep nuclei, 3 on each side, white matter, and then a surface cortex of gray matter, which is sort of two-toned. To the right here we see a cat cerebellum with just a cellular stain. And so here are the cell bodies of a nucleus. This is white matter, and then here is a cortex, and the cortex is clearly two-toned. There are these small, dense population of small neurons, and then there's a relatively acellular area on the surface, and in between, which you cannot see at this magnification, there are the cell bodies of the Purkinje cells. And so these 3 neurons form a circuit: the granule cells, synapse on dendrites of Purkinje cells, and axons of Purkinje cells, synapse in the nuclei. And so during development then, this circuit has to be formed. The cerebellum develops bilaterally from rhombic lips, which are expansions, dorsal expansions of the alar plate. As you can see down here, these rhombic means hind, and these are lips that grow up from the hindbrain. They will meet at the midline, creating a vermis and bilateral cerebellar hemispheres. Here you see the cerebellum growing up from the hindbrain region. Also, some of the neurons in the rhombic lips will migrate out of the cerebellum, migrate to the ventral pons, and become pontine nuclei, which will send their axons into the cerebellum. So next we want to consider the cellular migrations that give rise to the circuits among cerebellar neurons. Let's begin by considering the circuits in a mature cerebellum. In the granule cells, the small neurons of the cerebellar cortex send their axons into the molecular layer, where the axons run longitudinally and make synaptic contact on the dendrites of the Purkinje cells, shown in blue here. And the axons of the Purkinje cells then go down and synapse on the nuclei. So we have three neurons that are forming a circuit. The granule cell synapse on Purkinje cells, and the Purkinje cells synapse on the deep nuclei. The gray matter of the cerebellum is going to come from the mantle layer, that is, the cells that are surrounding the fourth ventricle in this case. And what we see is, first of all, superficial and deep cells that you see here in the rhombic lips. And these are going to give rise to the Purkinje cells and the neurons of the deep nuclei. So you might think that the more superficial cells here would migrate to the cortex and become Purkinje cells, and the deeper cells then would become nuclei. But it's just the opposite. The deeper cells of the mantle layer are going to become Purkinje cells, and they will migrate through the more superficial cells and then become Purkinje cells. And as the cells that they migrate through will remain deep and become the nuclei. So in this process of migration, Purkinje cells establish contact with neurons of the deep nuclei. And as they migrate, that contact is maintained by elongation of the axon. It's this process then of migration that establishes contact. Those neurons that fail to establish contacts and get neurotrophic molecules, those neurons will degenerate and disappear. The ones that have established contacts will persist as Purkinje cells in the cortex and deep nuclei. There is another population of neurons that form an external germinal layer. Here we see neurons migrating as neuroblasts up to the superficial surface. Here you see them here in red. And these neurons then are going to migrate through the Purkinje cell, through the molecular layer, and become granule cells. So here you see them migrating through. And here we see these cells then that are going to migrate through the Purkinje cells. And as they migrate through the Purkinje cells, they establish contact with Purkinje cells. And so it's the migration of neuron populations past one another that enables them to establish connections. These connections then create the circuits that we see in the adult. The midbrain, which is shown here and enlarged here, undergoes less elaborate differentiation than the forebrain and the hindbrain. The cavity of the midbrain is the mesencephalic aqueduct. It is an aqueduct and not a ventricle, because it is completely surrounded by neural tissue. Ependymma is lining the mesencephalic aqueduct, but the pia mater is out at the surface, and so there's no opportunity to form an ependymal pia layer, which could give rise to a choroid plexus. Every ventricle must have a choroid plexus, and in the absence of a choroid plexus, though it's called an aqueduct, alar plates of the midbrain form colliculi. These are rostral colliculi. These bulges are reflex centers for vision and caudally for hearing. The basal plate gives rise to two cranial nerve nuclei, the oculomotor, which you see here, and the trochlean, which is located more caudally. The midbrain is the most rostral extent of the basal plate. The entire forebrain will not have any efferent neurons because it lacks a basal plate. You can also see that there's a mixture of gray and white matter. The forebrain, which is derived entirely from the alar plate, gives rise to the diencephalon and the telencephalon. The diencephalon has a neural cavity that becomes a very narrow third ventricle. The roof plate of the third ventricle gives rise to bilateral choroid plexuses that secrete cerebrospinal fluid. The floor of the diencephalon gives rise to a neurohypophysis for the pituitary gland. The mantle layer of the diencephalon gives rise to gray matter, the thalamus, hypothalamus, etc., that surrounds the third ventricle, which is narrow in the center. It is significant that the optic nerve and retina develop as outgrowths of the diencephalon. So here we see a stalk growing out, forming an optic cup, and here we see a section through an actual optic cup. the stalk, the optic nerve. The optic cup gives rise to the retina. The thickened region here is the optic part where the rods and cones and other cells that process vision take place. This outer border becomes an external pigmented layer of the retina. And then from the surface, a lens is formed from a epithelial plaque coat on the surface. This image is a transverse section through an adult dog brain. It shows both diencephalon and telencephalon. This region here is the diencephalon. It features optic chiasm ventrally. Here is a third ventricle. It's obliterated in the center by thalamus, which is grown from right and left to obliterate the center. But here's the third ventricle up here. It has choroid plexus and a thin roof. This is also third ventricle here. This region is the hypothalamus below the thalamus. This also shows the telencephalon with bilateral cerebral hemispheres. In the telencephalon, we have a lateral ventricle. Each has a choroid plexus, which actually communicates with the choroid plexus of the third ventricle. And then we have gray matter, which is forming a surface cortex, as well as deep gray matter, which are called basal nuclei. The telencephalon, or cerebrum, consists of bilateral cerebellar hemispheres, which are formed by outgrowths of the forebrain. So here we have bilateral outgrowths of the forebrain forming right and left cerebral hemispheres. And if you take a cross-section through this, you would see something that looks like this. So we see that the third ventricle communicates with the lateral ventricle. Thin, stretched roof plate forms the choroid plexus bilaterally. Here we see that the third ventricle communicates with each lateral ventricle via an interventricular foramen. Lamina terminalis forms the rostral wall of the third ventricle, the end of the original neural tube. Meanwhile, the mantle layer of the telencephalon will form basal nuclei, which remain in the vicinity of the ventricle, and also will send neurons out to the surface to form a cerebral cortex. Here is the cerebral cortex of a cat. Here it's enlarged. So there are defined 6 layers of horizontal layers of cerebral cortex. And all of these cells migrated from the mantle layer to the surface of the cerebral hemisphere. They were guided by glial cells from the region of the ventricular surface to the outer surface. The migration occurred in waves. First wave that arrived at the cerebral cortex was not the most superficial layer, it was actually the deepest layer. Then a second wave migrated, and that passed through the deepest layer to become the next deepest, the 5th layer. Then the next wave of migration passed through the 6th and 5th to become the 4th layer. The 3rd layer passed through all of these to become 3rd and the 2nd. And so the cerebral cortex was formed by waves of migrating cells. And these cells established connections as they migrated through the cells that had arrived earlier. And so cell connections are established within the cerebral cortex by waves of cells migrating through previously migrated cells. And those cells that were able to establish connections and get neurotrophic molecules survived. Those cells that were not able to establish connections as they migrate through subsequently degenerated. And this process of forming connections creates columns of connected cells which become the functional units of the cerebral cortex, where all the cells within a column are connected to one another, and all of these respond to some feature of a stimulus as the functional unit of the cerebral cortex. It is worth noting that nervous system development continues in the neonate. Doesn't stop at birth. And that's particularly true of carnivores. Carnivores are born pretty helpless, contrast, herbivores are born much more mature because they have to be running in a matter of hours. But in both cases, the nervous system development will continue into the neonatal period. And some neuroplasticity will persist throughout life. This cartoon shows how two very different arrangements come from the same mantle marginal layer. So in the case of the spinal cord, there's a great need for fibers and not as much need for cells, and so the axons are put on the outer surface where there's greater volume. The process resembles the original arrangement with the ependymal cells, gray matter, and white matter at the periphery. In the case of the forebrain, there's a great need of cells, and so the cells are put on the surface where there's a great volume to accommodate all of those cells. Some cells remain in the mantle layer region by the cavity. These will be basal nuclei, and these cells will be cerebral cortex, separated by white matter. This is achieved from the same starting point, with the mantle layer giving rise to migrations of cells to the periphery. And so this creates the cortex at the outer surface, separated by white matter from cells that remain in the original mantle position. And so this concludes this nervous system development screencast dealing with central nervous system regions.